So in this video, we're going to think about control of the eukaryotic cell cycle and how cells are able to make decisions at a molecular level. So let's starting off about thinking about how cells divide and the processes that go on. So let's start, this is in eukaryotes, it's different in prokaryotes. But if we think about a eukaryote that is dividing, that has sexual reproduction, we'll start with fertilization of the egg. So when sperm meets the egg, obviously that is going to then trigger a new set of cell divisions. So we have a cell division cycle, which we'll put all the details of in a minute, but we've got a cell division cycle there. So in early development, we'll just be going round and round that cell division cycle quite a lot. But then what we can have is exit from the cell cycle to have cells that start to specialise and differentiate. So we have cells go into what's called a G0 phase. So these are non-dividing cells. And those cells can go on to differentiate, which means to specialise. Okay, so we've kind of got a balance with cells. You can either be dividing and relatively non-specialised, or you can stop dividing and you can specialise or differentiate. So something like a nerve cell would be an example of a cell that has stopped dividing, but it's highly specialised and it's highly differentiated. You kind of can't do both. You've got to do one or the other. Okay. So we've got fertilisation. We can go through cell divisions. We can go around that multiple times. And the sorts of cells that do that we usually refer to as stem cells. So stem cells are dividing. We might then have a choose to exit and go into this differentiation pathway. The other thing that might happen at each of those stages is to have cell death. So you could have cell death as a result of something going wrong in the division cycle, or you could have it from either of these stages. And the formal name for cell death in animals is apoptosis. Okay, so this is controlled cell death. The cell's very much in control of it, but sometimes the cell makes the decision actually the right thing to do is to kill off the cell. So for example, in the development of your hands, you actually start with a hand that starts as a paddle. So very early on in development, you have a hand that looks like this. And then you have cell death happens in a controlled pattern to make your hand with 5 fingers. So it's not that the fingers grew out, it's that the cells were killed off through apoptosis. So we had apoptosis happening in selective regions on the embryonic hand, and that's where your fingers come from. So it's cell death between your fingers that gives you the shape. Okay, so that's the overview of the sorts of things that cells can do. So let's have a think about this cell cycle in a little bit more detail. Okay, so this is this cell division cycle here. Okay. So the cell division cycle is divided up into 4 phases. Okay, so we first of all have a phase called G1, which is a growth phase. We then have a phase called S, which is for DNA synthesis or replication. So if you've thought about DNA replication, thought about the opening up of the bundle, leading, lagging strands, Okazaki fragments, all of that stuff is happening in S phase. That's we duplicate the genome. Okay. Then we have another phase, which is G2, which is another growth phase. Usually your organelles will reproduce in G2 phase as well. So you need to make new mitochondria. If you're a plant, you need to make new chloroplasts. And then we have our final phase is M phase, which is mitosis. So all of anaphase, telophase, metaphase, all of those sorts of things are happening in mitosis. The rest of it is interphase. Okay, so all the process of mitosis is happening then. So you'll notice that DNA synthesis and mitosis happen at opposite sides of the cell cycle. So if you were taught that the first thing to happen in mitosis is DNA replication, that's not true. They're in completely different bits of the cell cycle. And then, as we say, we have these G0 phases, so you can go into G0, which is the non-dividing, or sometimes called quiescent cell phase. You can only do that from G1. Okay, so you can go from G1 into G0, but you can't go into G0 from any of the other phases. Okay. So those are the phases, and obviously the cell needs to have really good tight control over this. You don't want to be getting these phases muddled up. You don't want to be starting to divide while you're still replicating the genome, for example. So the cell has really tight control over the cell cycle. It does this with two types of proteins. Okay, so we have two proteins, which I shall draw as such for the rest of this. Okay, so we have two proteins. One is called a cyclin, and one is called a CDK, which stands for cyclin dependent kinase. Okay, and you should know that kinases are proteins that phosphorylate other proteins. Okay, that's what kinases do. They go and phosphorylate other things. Okay, so that will phosphorylate target proteins is the job of the CDK. Okay, the cyclin has no enzyme activity. It doesn't actually do anything. But what it does have is binding sites, so it controls specificity. So the cyclin tells the CDK which proteins to interact with, which ones to phosphorylate. And the cyclins are named because they have very characteristic gene expression patterns. Okay, so what happens is if we measure the production of protein across the cell cycle, so if this is protein, so we've got G1, S, G2 and M, then what we find is that the cyclins have these kind of rhythmic patterns of expression. Okay, so so we have, for example, there's a cyclin called cyclin B is expressed at the end of G2 and into M phase, but we've got other cyclins there. So cyclin A is associated with S and G2 phase. We have cyclin E, and we have cyclin D. Okay, so different cyclins are expressed at different times in the cell cycle, and because the cyclins have the binding sites, they can control the specificity. That means that at different times in the cell cycle, we're targeting different types of proteins. So the cyclin is going to tell the CDK where to bind with and what to phosphorylate. Okay, so the CDKs they don't have these rhythms of expression quite so much, whereas the cyclins are very strongly associated. You only find the proteins at particular times in the cell cycle. Okay, so let's think about one of these points a little bit more detail. So what I'm going to do is to think about the checkpoint that happens at the G2 to M phase transition. Okay, so once you've decided to go into mitosis, that's a pretty irreversible decision. You're going to start to pull your chromosomes around, you're going to divide the cell. You really want to make sure you've got that right. So you want to double check that the genome has been replicated properly. You've got additional copies of all the chromosomes. You want to make sure you've got enough mitochondria. You want to make sure you've got enough energy in order to go into that M phase transition. So the G2 to M phase transition is a really critical point of control within the cell cycle. So we're going to think about how this works in a little bit more detail. Okay, so let's first of all, I'm just going to draw out the phases. So we're going to think about G2 going into M phase, and then obviously it will start a new G1 phase. Okay, but it's really, it's this checkpoint here that is really important. You want to make absolutely sure that you're ready to start going into M phase. Okay, so how does this work at a molecular level? Well, we need to think about our two proteins. So we're thinking about CDK, and it's CDK1 in this case, and it's cyclin B are the ones that are involved in this transition. So let's build it up. So let's think about G2. So if cyclin CDK, cyclin, I'm doing CDK1, cyclin B, okay, is the complex. And that's going to activate all sorts of stuff to do with mitosis. So it's going to activate condensation of the chromosomes, breakdown of the nuclear envelope, all of those sorts of things. Okay. So let's think about both of these proteins and what they're doing at these various different stages. So let's first of all think about the beginning of G2 phase. Okay, so the start of G2 phase, let's just pop the end of that arrow in. The end of G2 phase, the CDK is basically found all the time. Okay, so this is CDK1. That protein's there all the time. Okay, it doesn't change its expression level very much. Okay, but without its cyclin partner, it's inactive. There's no cyclin. Okay, so not much is happening with CDK1. It's not targeting anything else. Okay. If we think about the end of G2, so during G2, we get the production of cyclin B. So the cyclin B gene gets switched on. So we start to make cyclin B. So by the end of G2, we will have lots of CDK1 and lots of cyclin B. Okay. But it's still inactive. We're not ready to go into M phase yet. We're doing our final checks. Okay, so we need a mechanism to stop that protein complex being active. It's not quite ready yet. Okay. So what happens is there is an inhibitory phosphorylation by another kinase called WEE1. Okay. So another protein called WEE1 has stuck a phosphate group onto CDK1 and it actually sticks it on the active site. Okay, so we are still inactive because of phosphorylation by WEE1. Okay, it's the WEE1 is named just for it's a WEE protein. It's from the Scottish, so the cells that have defects in WEE1 just look much smaller. Okay, so at the end of G2, we've got the cyclin and we've got the CDK, but it's still inactive because there's that phosphate group there. Okay, so then we go into M phase proper. Okay, so now we need the whole thing to be active. Okay, so we've now got the CDK, we've got cyclin B, and what happens very quickly and very abruptly is that phosphate is removed by a protein. It's a phosphatase protein called CDC25. Okay. So the removal of that phosphate now means that we are active. So we're going to go phosphorylate target proteins. Okay, so we've removed the inhibitory phosphate, we've got the CDK, we've got the cyclin, everything is now active. Okay, so we can now start mitosis. Okay. But there's another thing that needs to happen is we want to make sure that once we've started mitosis once, we definitely don't want to start it again. So what the best way of doing that is exactly what the cell does, is at stage 4, again we've got the CDK, is we start to destroy cyclin B. And that's by a big complex in the cell called the proteasome. Okay, so we don't need cyclin B anymore. We've initiated mitosis. It's done its thing. It's all gone fine. So what we really don't want to do is start mitosis again. So what we're going to do is to make double sure we can't start it by destroying cyclin B, and then that means that we are then inactive because we have no cyclin. Okay, so we kind of reset the system. We started off with no cyclin. We made the cyclin, but we had this phosphorylation to keep it inactive. We then got rid of the phosphate group, so we had the active complex. And then one of the things that it targets is the proteasome, so it catalyzes its own destruction at that point. So this is quite a nice example of we've got both transcriptional and post-transcriptional control going on here. Okay, so if we just draw, again, we draw a graph. Okay, so we've got G2, M, and G1. Okay, we've got 3 different things going on here. Okay, so if we think about the CDK, that's there all the time. That's a constant. Okay, so the protein is always there. So that's not being sort of regulated at the level of gene expression, but our cyclin is. So at G2, we start with low levels of cyclin B. We go up to high levels of cyclin B that we keep through the beginning of mitosis, and then we start to destroy it again. Okay, so that's the cyclin B protein. So this bit here will be gene expression. And that will be destruction. Okay, so that's controlling how much protein we've got, but we've got this additional thing of we don't want a very gentle increase in the activity, we want a really abrupt piece of change in activity at the checkpoint. So if we do the activity graph, we'll have very, very low through G2, very, very low, then suddenly at M phase it will go up, it will stay high for a while, and then it will start to decrease again. So this is CDK1 cyclin B activity. So we've got a different shape, and that very abrupt transition there is a result of post-transcriptional regulation. So if you remember, gene expression is generally quite slow, it's quite gradual, whereas if we have post-translational or post-transcriptional mechanisms, in this case by phosphorylation, then we can have very quick regulation of the system. So if we just go through that again, so G2, we're still growing, we're still checking that the genome's been replicated correctly. CDK is there, not active because it's not got any cyclin. The cyclin starts to get made, but we have this additional inhibitory phosphorylation by the Wee1 kinase, so it's still inactive. When we're absolutely sure the genome is ready, everything's ready, that we can go through mitosis, then very, very quickly that phosphate gets removed. So the whole complex is active. It can go and phosphorylate target protein, so the nuclear envelope starts to degrade, the chromosomes condense, and then once that's happened, cyclin B sets up its own destruction, so it activates the proteasome, so we destroy cyclin B and we go back to our low levels of activity. So this is an absolutely classic example of how cells are able to make decisions at a molecular level. We've actually not got very many proteins, but they're controlling a really, really important aspect of the cell division cycle.